All right, so I'm back. Cleared up some computer issues I was having on the server, um, and so we're kind of ready to go. Again, I have a Trinity assembly made, and so we'll kind of see what we do with that downstream. Right, once we have these reads assembled, once we have a transcriptome assembled, we're just going to kind of take a tour of the main output, and then how we can deal with it, right? What kind of comes next? All right, so I have all this done in a FastQ, my FastQ folder. All I have are my clean reads and just the Trinity output dir. So normally when this is all done, you'll have this file, this folder, and it'll be populated with a whole other lot of files. And a lot of them are massive, and most of them are not necessary. So I've already gotten rid of all that garbage. So when we look at my Trinity output dir, all that's going to be here is the Trinity fast. Now a couple of things ignore. So ignore these two file directories. I'll kind of cross that bridge in a second. So right, again, I've gotten rid of everything else except for the raw assembly. It's good because honestly, this is all that really matters. Now, some other downstream programs can use something like a mapping file. I haven't taken a look at that exactly. I may do so here in a minute, but not necessarily today. It's not necessary. Let's just say that. It's an optional file, right? So if there's really two files to matter, it's the trinity.fasta and then maybe like a mapping file, but whatever. We're not there. This is just kind of like basic stuff. All right, and so we have this Trinity output. That's the main one. That's all that we care about. And it's reasonably large, 225 megs. And if we look through this, so we're just going to kind of take a tour of this output file. So let's Trinity. And so what we have here is going to be, it's a FASTA file, right? It's a FASTA format. It just tells us, it gives us a transcript name and how long it is in base pairs. And a couple other things not that important, right? So we see a whole lot of different sequences and no annotation. So we have no idea what these transcripts are related to, what genes they're derived from, where they come from the genome. No idea any of that stuff. And so this is reasonably long. And so if we count how many sequences are here, just using a grep-c on the open caret, 255,000 transcripts. And so as I've kind of mentioned before, like on average, there are about 10,000 genes expressed in any given tissue. It seems to be, at least in a vertebrate, it averages out about 10,000. This is way more than 10,000. So there's a lot of stuff in here. So we for sure know that there's going to be true genes in here. There's probably going to be some long non-coding RNA stuff in here as well. And there's going to be artifacts, right? So assembly is a good solution using these small reads, but it's not, I wouldn't exactly say it's a solved problem. We can use this information and it's way better than nothing, but that doesn't mean that this is perfect, because there can be chimeras in here. So there can be genes that kind of get merged together as one, right, as a chimera. There can be assembly artifacts. There can be multiple transcripts for each sequence, right? So we also have to recognize that there is alternative splicing, and alternative splicing is pretty common, right? So you'll never, in a good assembly, you'll never see just like 10,000 genes and we're all said and done. We have to look through this. We have to filter out what we want from it, and kind of get rid of the rest. So let's see. So kind of the next step, once we have this Trinity file, the next step comes to be annotating it, right? Because again, if we just look at this, all we see are just sequences, transcripts, right? Each sequence is its own unique transcript. But again, we may see multiple transcripts for a gene. And honestly, we can sometimes the exact same transcript will be assembled more than once, right? It does happen. I know it happens. All right, so these are all varying of lengths, 400 bases, 200 bases, right? A lot of this stuff is going to be garbage, or at least not necessarily garbage. It's probably functional. It's probably there are RNAs in here, but if it's not mRNA, it's not going to help us out all that much. So how we annotate genes is to find open reading frames, right? The lowest level solution is to find an open reading frame. And so an open reading frame is just methionine to stop codon. And so in that frame, there should be only there should be a start codon and only one stop codon. But after that first stop codon, then there should be a bunch because we're out of frame, it's not being coded, whatever. But there should be a very long stretch of amino acids or codons that will stretch from a methionine to a stop codon. You know, plus the UTRs, right? There'll be a 5' UTR, start codon, open reading frame, stop codon, 3' UTR, right? That's how an mRNA transcript should look. Right, so even here, this might be lucky. This is probably just, this may or may not be actual, because we don't have the reading frame, right? We don't know where the reading frame is. We don't know where the reading frame is, but nonetheless, this transcript does start with an ATG. It's probably not our true start codon, but that's what we're going to look for. And believe it or not, I think even finding open reading frames in transcript data is kind of tricky. And it's tricky for a couple of reasons. One, not all these transcripts are going to be complete, right? We may only see a partial fragment that may be completely internal. So we don't actually see the start codon, we don't actually see the stop codon. We just see this long stretch of amino acids that could fit within an open reading frame. It's just we don't see a start or a stop codon, right? It'd be an internal fragment. We can see fragments where we only see a stop codon, right? We'll have an open reading frame and then a stop codon and then garbage after that. And then another problem is that not all genes start with ATG. Methionine is not always the start codon. Like when we go through intro biology, I say it, right? All genes start with ATG. Dirty secret, that's not always true. Not all genes start with an ATG. And so what do you look for? How do you look for the start of a gene if they don't all start with ATG? So finding open reading frames is tricky. I have made programs to do that, but it's more or less just looking for the distance, like translating in all 6 reading frames and trying to find the longest region between stop codons, right? That's the best I can do. That's the absolute best I can do, is try to find the longest region between 2 stop codons. Even that may not actually be the reading frame. So it is a tricky problem, and the best solution that I know how to deal with this is to use this program called TransDecoder. If I can't make anything better than I'm just going to use what's something already on the market. And so these 2 output files, these 2 output directories were made by TransDecoder. And so I have TransDecoder also hanging out in the opt, right, along with Trinity and Trimomatic. I also have TransDecoder hanging out in the opt. And so TransDecoder has a couple other, has a few modules. It has TransDecoder long ORF, TransDecoder large test, and TransDecoder.predict. For this, when we're trying to find the open reading frames and all of this stuff, it's going to be TransDecoder long ORFs, right? So let me clear this out and act like I'm going to run this program. I'm not going to run this program because whenever I ran it, it took about 10 minutes to finish. So you're not just going to watch me sit here for 10 minutes. I've already generated it. But notice, if I'm going to call this program, opt trans long. And then if we look at the options, right, we'll see some of the options here. The only required option is the -t. And so that's going to be our trinity.fasta. So -t is our only option that we have to care about. Again, we can have this gene trans map that's also made by Trinity. I'm not going to use it. I've deleted mine. I kind of forgot this even existed before I used it. And I don't honestly know what's going to happen if I did call this option. I may explore. So if y'all have made a Trinity FASTA, but you haven't deleted everything yet, leave me a trans map file and I may use it. Just to explore and see what happens, right? See what the difference is between a raw transdecoder analysis and one where we have a gene trans map. The M can be modified, so that's just saying the minimum protein length, so the number of amino acids that will be translated. So the reading frame has to be 100 codons. If it's shorter than that, get rid of it because it may not be true, it may not be real, or it's just going to be so short already it's not going to help us in any way whatsoever. So the default's set to 100 amino acids. If anything shorter, we just get rid of it. Don't worry about it. We can set the genetic code, but honestly, if you're for most transcriptomes, you're going to use the universal, right? I mean, the same genetic code is the same genetic code, regardless of what nucleus you're really looking at. So that's fine. And strand specific, not going to worry about that. I wouldn't even call that. What that's saying is like if we have our transcripts, only look in the forward-facing reading frames, right? Don't look in the reverse-facing reading frames. Only look in one direction. I don't want to do that. I'm going to look in all 6 reading frames, so I'm not going to call that. And output dir if you want it. Yeah. And the genetic code, again, is just universal. So if I'm running this program, again, for real, all I'm doing is calling the long ors, -t, and then trinity.fasta. Right, that's all it takes. That's all it takes. And again, this takes about 10 minutes to run, so I'm not going to run it. And I don't want my stuff to get overwritten, so I'm not going to run it. But nonetheless, it will produce a couple of directories. A transdecoder dir, and then like a checkpoint longest. And I think the checkpoint's there in case your job gets interrupted. You can restart it, and it will start from where it left off at, not all the way from the beginning. Right, so that's there if we wanted it, but Trinity.fasta transdecoder is going to have our stuff that we care about. Okay, and so some of this is made by me. That's made by me later, but all the time you'll get these four files. You have a base frequencies out file, and then these 3 input or output files. That's going to be the CDSs. The PEP, which is the protein, the amino acid sequence, and then a GFF. If you care about the coordinates, if you need the coordinates for whatever reason, they're there in a GTF or a GFF. Right, so the base frequencies ultimately gets used. Oh, that's almost equal. That's nice. That's almost equal. But the base frequencies get used to annotate open reading frames. So I think there are some nucleotide biases. I hope I'm not wrong, but I think there are some nucleotide biases in open reading frames or in coding sequences than outside of a coding sequences. And so it kind of will use that information to help identify reading frames. It does some other magic. Not going to worry about exactly what it does. It does it better than what I can do. Again, we're identifying open reading frames. And so it returns all of the sequences where it found a past open reading frame given the conditions. So we'll look at the CDS first because that's just going to be the DNA open reading frame. So we'll look at that first. Longest or CDS. Okay, coding sequences. And so each of these FASTAs now gets annotated, right? So we have the transcript name. And now all of this stuff was made, this header information was made by TransDecoder. And so it's saying this reading frame is internal, which means it found a long string of codons, but it couldn't find a start or stop codon. So it's probably, it's a fragment. And it gives us the orientation, right? It gives us a positive orientation. It goes in the forward direction. That's also internal. Now this is a little silly. This confused me for a while. It still kind of confuses me. But it's saying it's a 5' partial. But in reality, it means that it's missing the 5' region, meaning that we're missing a start codon. But we do have a stretch of all of these would be in the same reading frame, and it ends with a stop codon, right? TA is a stop codon. It also gives us a 3' partial. And so a 3' partial is the opposite. It means that the 3' area is missing. And so it starts with an ATG, right? It starts with methionine, but it never found a stop codon throughout the transcript. So it gets labeled a 3' partial, meaning it's missing a 3', it's missing the stop codon. And then lastly, so we have the internals, we have the 5' partials, 3' partials, and lastly, we will have the complete. I'm going to have to look for a complete. Right, there's a complete. And so it at least is saying that it found a start codon, right, ATG, found a start codon, and it found a stop codon. And there was a whole open reading frame between those two positions, or those two codons. So we get a complete annotation in here. Right, so we get these four: complete, internal, 3' or 5' partials. And a lot of these are internal. A lot are internal. A lot are partials. So let's first off see how many passed. Like how many transcripts actually had an open reading frame. And again, we're just going to grep. I'm going to just do a grep-c on that, on the caret. And so I'm just going to do it on the Trinity FASTA so we know what our starting reads were. So we had 255,000 transcripts to sort through, and we found, let's see, 181,000 open reading frames. Okay, and so of those, we may ask how many were complete, how many were partial. In which case, we'll just grep on the word. So I can grep complete, and it's gonna just give me the headers, right? I'm just gonna let that go. Right, it gives me all the headers. So I can just WC that real quick. I can just grep C on that. Grep C complete. And so that of the 181,000 transcripts or open reading frames that were annotated, only about half of them are complete. But it's still way more than the number of transcripts we'd expect. Honestly, if I were looking at a transcriptome, given about 10,000 genes, given all the alternative splicing that can happen, about 100,000 transcripts that link back to genes is about what I'd expect. Doesn't mean that there's probably more. Again, there's artifactual stuff in here that we have to deal with and get rid of. But at least it tells us that half of them are complete. But we can certainly, grep on partials to see how many are partial. 60,000, and internal. We can grep on internal to find out 21,000 were internal, right? All right, now what? Now we have the open reading frames, but we still don't know what genes these came from. And just so we see it, right? Let's look at longest ORF path, right? So now these are the full translated amino acid sequences. Right, so what TransDecoder does is it more or less, it just returns, it gives me that transcript back, but it also says I only want the open reading frame, right? So the UTRs, the 5' and 3' UTRs, I don't want them, I don't care about them, if they're annoying to deal with, just get rid of them. Just give me the open reading frames, right? So we get the CDS open reading frame, and we get the translated amino acid sequence reading frame. And so you can clearly see like the 5' partials. You know, they don't have a start codon, but they end with a stop codon. Right? They don't start with methionine. You see the 3' partials start with methionine, but they don't stop with a stop codon. Right? So it's the same information that's in the CDS. It's just all been translated. Internal, a lot of internals. 5' internal. There's a complete one. That's going to be a short sequence. That may or may not be real. We'll see. Right, because that's only 150 bases or 115 amino acids. Most protein coding genes, doesn't mean it can't exist. Most protein coding genes are about 300 amino acids. Most of them. That, yeah. 5' partial, right? This is definitely part of a bigger gene. It's definitely part of a bigger gene. There's probably no denying that. Anyways, kind of the next step. So I do have this step already running in a tmux, but we'll see that I have this long orf blast out file running. So I have not talked about blast yet, but I'm going to demonstrate blast in a second. I'm going to demonstrate the properties of blast. I just need one transcript to look at real quick. Okay. I'm just going to look in the PEP. So yeah, I'm just going to look in the PEP for this gene. Okay, so here's our transcripts. It's internal, but nonetheless, we at least want to know what this is. And so we can use two things to figure out what it is, and it's basically a comparison search. So I'm going to grab this sequence real quick just to demonstrate this thing called BLAST. And so I think in genetics you're probably introduced to BLAST. I'm not entirely sure. You may or may not be familiar with BLAST, but what BLAST is a search algorithm. And it's more or less trying to find similarity. It's a similarity search algorithm. And this very well might be not the oldest tool. It's close. This was produced in 1991. And even though it's 30 years old, it's still probably the most commonly used program in bioinformatics. Right? Almost everything starts with BLAST. And almost any time we're going to annotate a gene, almost everybody will use BLAST to do it. You'll see some variation around it, but 95+% of the time, we're going to use BLAST to do this. And so what BLAST does is it's going to compare our sequence to stuff that's kept in a database for comparison to find identical sequences. And so this is run by the NIH. So this was developed, I think it was developed by the NIH. If not, it's been part of the NCBI for that long. You know, all that really updates in BLAST. Like BLAST really has not updated in 30 years. It still works effectively the exact same way. All that really has changed is just how we interact with it. And so I'm just going to take that sequence I copied and just run a protein BLAST on it. Right, because I have a protein and I'm going to compare it to a list of other proteins for what's the most likely protein that this thing came from. So I'm just going to copy that in here. I'm going to get rid of some of this stuff just to keep the header shorter. And we'll see what happens. I have no idea what's going to actually happen. I don't know. I have Danny over there. No idea what's about to happen. Hopefully we'll get a hit and I can kind of describe these hits. And no guarantee how long this will actually take. So I'm going to start it. It may take a minute. It may take 5. We'll see. But while that's running, I'm going to demonstrate another database that's very, very helpful to annotating vertebrate genes. And that's the SwissProt or the UniProt database. Oh, that was pretty fast. So that was done. Okay, yay. So it's saying so if we're looking at the NCBI BLAST directory or output, we see all these reds, and all these are effectively hits. These are BLAST hits. Okay, I don't know, I got a little freezy. I don't know if that's freezy on your end, but hopefully it's not too freezy. Anyways. And so what all this is like how close were these hits in the database to our sequence of interest. And so our first hit, our best hit, is this ANKFN1 isoform. And it's specifically from Pongo. And so there are a couple of pieces of information in here. Yeah, I think that's, yeah. That feels laggy. Blah. If there's really like all of this stuff, it's these two values, percent identity and the E value. Because what these are basically saying is an E value is like the probability that these two sequences are similar by chance. And so this is a 1 times 10 to the negative number. So basically the smaller the number this is, so as that exponent goes up, the less likely these two sequences are identical by chance. And percent identity is just how similar our sequence was to that hit in the database. And so the nice thing about this is we get different hits to different species, but it's all the same gene, right? They're all of this A and KY or ankyrin repeat. I don't know what this gene is, but this gene has been annotated. It's been annotated and everything. So right, our second hit is a human. That's what it came from. A third hit is a mouse. Another one is a chimp. Right, so it's very confident that this fragment of a gene came from this ankyrin gene. Again, don't know what it is. I just know that my gene is most similar to that. Okay. And so that's more, I mean, this is a very simple idea of how BLAST works, and we can certainly go into a lot of more details into it. But the point being is that we can at least visually see what BLAST is doing, right? We can give BLAST a sequence, get a result back, and it tells us if it's known or not. At least tells us if it's known and how likely it is, it belongs, it's a homolog or an ortholog of the hits. And again, the main one, probably the most important number is the E value, right? This is 1E, 1 times 10 to the negative 96th. That's a pretty small number, like 95 zeros in front of a 1, right? Really small decimal. So it's most likely this ankyrin gene. It's very unlikely that my sequence was identical to the ankyrin's by chance. Anyways, I'm gonna get rid of that. And so the other thing was the SwissProt database or the UniProt database. And so UniProt is awesome because not only can I identify a gene, it's going to give me a lot of functional information too. So let's see, let me grab that real quick. So let's say, so this was my hit, and it's from, let's see, let's just see what an ankyrin human looks like. At least let's see if SwissProt will tell us our functions. So I'm just going to search for that gene, and we're going to see what happens. Okay. Annotation's kind of okay. It's all right. It's not great. Not a lot of functional information in it. right? Because a good hit will say like tissue it's mostly in. Expression is buccal mucosa cells, and it's effectively everywhere, right? It's constantly expressed. If it's in 118 tissues, it's constantly expressed and everywhere. But we don't get a lot of functional information in this. And it's just because it's kind of weakly annotated. But nonetheless, like this is kind of my go-to source. So if I want to know what my gene is, I'm going to blast for it. And if I want to know what the function is, I'm gonna use UniProt to do so, or SwissProt. Now the nice thing about UniProt is that we can download the entire database and use it for a blast search, right, as a reference. Because this whole database contains hundreds of thousands of proteins from humans, mice, rats, dogs, cats, horses, right? Almost anything that's reasonably related to a human that has been functionally annotated is going to be in the UniProt database. And so I've already gone ahead and downloaded the entire UniProt database. It's not all that big because honestly, annotating functions in a protein is very, very hard. It's very time consuming. And just because it's doing a function in one species doesn't mean it's doing the exact same function in a related species, right? Protein function changes among lineages a little bit. But nonetheless, I had this database downloaded. And so that's hanging out in opt as well. No, that's hanging out in data. That's hanging out in data where we've kept other things. So this is in SwissProt. Right, so this is the entirety of the UniProt database. It's probably a little bit bigger than what I have downloaded. This is an older version. I downloaded it last year. It's probably updated by a year. But nonetheless, we can see what's in it. Right, so it gives me these identifiers, these SP accession numbers. Like this Q is specifically linked to this protein in the SwissProt database. So if I search for this Q number in SwissProt, it should tell me more about that gene, right? It's going to tell me what species it came from, FRG3, frog virus. That's that's a transcription factor in frogs, poorly annotated. That's not a good example. Other virus things, other viruses. Let me find a human. There's a human. Beta alpha. That's probably, let's see what that is. Let's see what that is real quick. Protein level, gene YWHAB something. 1433 protein beta alpha. Right, but at least it tells us a little bit of information, and it's been experimentally validated. So there's experimental information behind this. It's reasonably well annotated. And so it can give us a little bit of information. We haven't really talked about these Go terms. It stands for gene ontology. But nonetheless, these are like words that get linked to a protein function, right? Enzyme binding, no, not very helpful, right? It just binds other proteins. Identical protein binding, it probably means it forms a dimer. It's a kinase inhibitor activity, so it's going to inhibit kinases which phosphorylate other proteins. It's a membrane organization for a biological process. It seems to be involved in plasm. Let's see where it's expressed. Not well, cellular component, right? So it's a nuclear expressed gene. No surprises there. You'll see them expressed in the nucleus, cytoplasm, right? Wherever it happens in a cell. Anyways, SwissProt, really, really awesome. And so, right, we're not worried about all the annotations just yet. We want to find out what our proteins are derived from. Right, so this is this is just my go-to database for annotating vertebrate genes. Because most likely, if it's closely related to a human, or if it's certainly a mammal, we're going to have pretty good information on it. 14 soybean, right? But there's still additional genes in here, right? That's from soybean, right? That looks like it's from tobacco. That is from tobacco. Anyways, so if we grep on the open caret, it's going to tell us there are about 500,000 sequences in here. Yeah, 561,000 sequences in here. All of them are proteins. And so we're going to find out what our proteins are derived from by doing a blast search. So I'm going to go back to my FASTQ folder, my Trinity out, specifically my Trinity transdecoder, right? Because that's what I have running here with this longest OR blast out. I have a tmux running blast. And so I have blast built into this. And so blast, we didn't really notice this, but blast has a couple of different flavors. Blast in is like a nucleotide to nucleotide search, right? We have a nucleotide query and a nucleotide database. So we might do a blast in that goes a little faster. Blast P is for a protein. That's where we have a protein query and a protein database. Protein query, protein database, and so we're searching proteins against proteins. Now blast X is if we have a nucleotide query but a protein database. Then we can basically tell blast to translate all of our sequences in reading frames and find the most likely protein. And that even gets crazier with T-blast in, T-blast X, but not all that important. We're just going to use a blast P. Now, even though blast might be the oldest program, it's not the oldest program in bioinformatics, but it's the most commonly used program in bioinformatics, the help menu is absolute not helpful at all. Right? So that's a help menu. That's a usage menu. Gives you all the options of what they can do and really has no good explanation of how to use them. Even if we do the help, I would not exactly call this a user-friendly and helpful database or helpful menu, right? It's long, it's a lot of options, they're poorly described, what they do, right? Sometimes, you know, but blast is awesome. Well, if you figure it out, if you know the right command line, it's awesome. And I'm going to tell you the right command line. So blast P. And right, the things that we need the most is this DB option. It's just so long. It's hard to find anything relevant. So let's just get cut to the chase. Blast P. I need a query. I need a query option. And for my query option, I'm calling in my query file, which is the file that I want to search through for hits, in which case that's going to be my longest orf.pep, right? Because I'm using blast P, I need to supply proteins. I need to supply my database that I'm going to use for comparison. And so that's hanging out in data. That's going to be the SwissProt SwissProt database. And then I have to kind of tell blast how I want this reported, because blast can give us like alignments, but it's not very helpful. But we can control how the output looks by using out format, out FMT. In which case that's going to be 6. Don't ask why, it's just going to be 6. This is a tabular output format. It's the easiest to work with when all is said and done. E-value, we are going to have a cutoff. Like, we don't want to return garbage hits. I at least want to return like okay hits. At least that's the best case scenario. You're going to return an okay hit. So I'm going to supply an e-value that if the e-value is bigger than this number, it's going to be disregarded. It's going to be ignored. And so traditionally, it's common to do like a 1e negative 10, right? So 1 times 10 to the negative 10th chance that this is equal. This is by random. But I'm going to do 1e negative 20. But this will go up all the way to negative 110 to the negative 100th and beyond. Sometimes it'll even just say 0, right? There's a 0 chance that these two things are unrelated. And so out format e-value, num threads, num_threads, can define how many processors we want to use in this job. It would be 6 processors. I'm just going to select 6. And the last one is max target seeks. And what this option is supposed to do is limit how many hits are actually returned, right? So if we look at the raw blast output from NCBI, it's a very, very long list. Well, if the top 10 all stay the same, I don't need that entire full list. Just give me the top 10. So I'm going to set max target seeks to, I'm going to set it to 10 in this example. So it's saying like only return 10 hits as opposed to all of them that you find. Sometimes, this is a little dirty secret about blast, sometimes if you running max target seeks 1, basically give me a hit back, right? If there is a hit in the genome, give it back to me. Now, we have previously thought that if you set max target seeks to 1, it's going to give you the best hit, but that is certainly not true. It's just going to give you the first hit it finds. So that can be a problem if you're not actually returning the best hit. Therefore, I usually leave it at about 10, and then from those 10, I'm going to use the best hit out of that to ensure that I've most likely found the best hit in my search. And then I'm just going to use my open caret to save. I would use my open caret to save that to some output file. In this case, I have it as longest orf.blast.out. And as I said, I'm already running that, so I'm not going to run it again. And I run it under these conditions. I think the only thing I changed was using a max target seeks of 20. But to make that go a little bit faster, you can reduce it to 10. But again, just because of the sheer number of people that are using this server, don't go beyond 6 threads, even though it will take a long time. Right, I have been running this for a little less than a day, and at this point, I have results. I'm going to use my cut and unique. Right, so I can let me just head that real quick. Head longest orf, right? And so it's giving me the transcript. It's giving me the transcript for that open reading frame. It's giving me a hit for that open reading frame. This first number is percent identity for this search. So it's saying they're 100% identical. And it was for 142 amino acids. And our e-value is 5.76 times 10 to the negative 96. And this last number is a bit score. Sometimes if there's a tie among e-values, because sometimes e-value will be 0. And so if you have like 5 hits that are all e-value of 0, you can still pick the best one by using the biggest number, this bit score, right? It's still probably the best hit of them all. Okay, and so let's say for this transcript, it gave me the same hit twice, one for a human and one for a mouse. And this transcript is more similar to a human transcript than it is to a mouse, as it should be. Not always going to be true, but it should be. E-value is times 10 to the negative 118. So, right, this is most likely a PCX2 gene, and etc. As I said, this is still running. And at this point, I can cut on the first column, look at the unique ones and count them, see how many are actually unique, see how many have actually already been done here. So cut dash F1, longest, or blast, unique, and I'm going to WC that. In which case, it's going to tell me I have processed 7,800,000 transcripts, or 78,000 open reading frames. And after that, it's just, I'm a little more than halfway done. I'm pretty sure I'm more than halfway done. This will probably finish today. But yeah, this will take a long time. By running only six processors on this, it's going to take a while. If I were by myself, if this were the summertime and nobody was on this, I would certainly use like 20 to get it done and over with in like 3 hours. But we don't have that I don't have that luxury, and we all need to be considerate about each other's use. So it's going to take longer, but everybody can go at the same time. But yeah, whenever that's all finished, we'll kind of talk about what happens next. But again, now we've gone from simple reads to fully assembled transcripts, and not just fully annotated transcripts. We know what genes are present. Now we know what genes are present in all of this. So that's kind of what's happening in all of this stuff, and we'll see what the next steps are. Probably Wednesday. I think Wednesday I'll be live on YouTube, but Monday I'll probably just talk to y'all privately. So until then, I'll see y'all later.